Hello, my name is Jacqueline Payton. I'm at Washington University in St. Louis. I'm an associate professor in the Department of Pathology and Immunology. And I would first like to thank the organizers for the invitation to speak in this conference. I'm really looking forward to sharing some of our work on RSV. And the title of my presentation is Viral Hijacking, How RSV Wrests Control of the Host Immune Regulation. First, I want to just preface this presentation by saying that my lab focuses on multiple areas of research, of which pathogen-mediated disruption of host immune response is one. We also spend a lot of time working on cancer and lymphoma, but obviously that won't be the topic of today's presentation. Today, I'm going to focus on our work on hRSV and how the NS1 protein disrupts host immune response gene regulation. So to begin broadly, there are a number of epigenetic changes that occur in response to infection. And epigenetic changes refer to a broad set of regulatory mechanisms that affect gene transcription. And so in the center, you can see a diagram of a gene with the arrow pointing, the black arrow pointing towards it, indicating transcription of that gene. And on the left, there are a number of different types of infection depicted in cartoon form, and then influences on the host, that infection, and also the environment, and of course, host factors such as signaling molecules. These all interact with the host's own genetic variation, which would be differences in genetic sequence in that host, and then epigenetic variation, which include modifications to the proteins that DNA is wrapped around, so histone proteins, DNA methylation, which is a modification directly on DNA, and then non-coding RNAs. These are all considered part of epigenetic variation. Transcription factors and transcriptional regulatory proteins are also considered part of epigenetic variation. All of these things contribute to gene expression, and these changes occur in response to infection as well. Now, viruses have a variety of strategies to try to disrupt host responses, including host epigenetic responses. And the type of strategy that a virus employs depends to a large extent on properties of that virus. So there are, of course, different genome types and replication processes for different types of viruses. For example, DNA, RNA, single and double strand viruses, negative and positive sense, whether they replicate in the cytoplasm or the nucleus, whether they integrate into the host genome or not. Then there are additional strategies that viruses employ that may or may not be related to those in the bullet points above, that they use for evading host immune response, including disrupting immune response gene expression. So genes that are changed in their expression in response to a viral infection and in some way contribute to the host immune response. In the following slides, I'll tell you a little bit then more focused about our study of the gene regulatory impact of one of these viruses, human respiratory syncytial virus. So human respiratory syncytial virus, or hRSV, is an important human pathogen, as many of you know. It's a common cause of lower respiratory tract infections in infants, the elderly, and immunocompromised populations. In other populations, healthy adults, there's also RSV infection, but it's a much more mild infection and typically does not involve the lower respiratory tract. Importantly, infection with RSV doesn't confer long-term immunity, and so an individual can be infected many times throughout their life. RSV can also predispose to asthma, which is a particular concern in very young people, so infants and young children. And recently, there's been fantastic developments of vaccines that have been approved by the FDA, but those have only been approved in older adults. So depending on an individual's risk factors, either 75 years old or 65 years old, and that leaves infants and children particularly still without a vaccine available. So I wanted to touch just briefly on the structure of human RSV and its component proteins, as it is then relevant for some of the slides that I'll talk about in a moment. So RSV is actually quite a small genome. It encodes for 11 proteins. And of these, we, in our work, have focused on non-structural protein 1, which is highlighted in the red box. It is the first gene that is transcribed in the RSV genome. And as its name suggests, it is a non-structural protein, which means you actually don't see it over here on the left in this diagram of the virion. However, in spite of the fact that it is not a component of the RSV virion, it plays important functions in RSV function and replication, infection. So to elaborate that a little bit, our collaborators in this work in the Lung Lab in 2017 defined the structure of hRSV NS1. And that is just shown here in a cartoon drawing of the structure of NS1. And they define some aspects of NS1 function that I will discuss a little bit later. But it has also been known from other investigators that NS1 is a known cytoplasmic immune antagonist. And note that I said cytosolic. There's quite a bit of literature on its role in the cytoplasm, acting as an antagonist, so blocking the host immune response. Also, notably, RSV replicates in the cytoplasm, unlike some other viruses. NS1 has been shown to be important also when other investigators deleted NS1 from the virus or created recombinant viruses that did not contain NS1 and found that those viruses were attenuated in their replication and also resulted in increased interferon beta mRNA levels, which makes sense when you think about NS1 being an immune antagonist, meaning that it would block interferon response. If you delete it, then there were increased interferon beta levels. There is also data to support NS1 suppression of interferon signaling via direct interaction with cytosolic interferon pathway proteins. And there is additional evidence that NS1 suppresses transcription of host response pathways, which is, of course, in the nucleus and not in the cytoplasm. But the underlying mechanisms of that are incompletely defined. And certainly when we began this project, there was even less known. My collaborators in the Lung Lab determined that NS1 has structural homology to RSV matrix protein, which is known to localize to the nucleus, which made us wonder, this was one of the first things that made us wonder about whether NS1 might have a role in the nucleus in gene regulation. So one of the ways that we determine how proteins function is by measuring their interactions with other proteins. And one way to do this is with a technique called affinity purification. And then, so in this case, again, my collaborator in the Lung Lab, Daisy Lung is pictured here, my graduate student, Nina Berry, former graduate student, Nina Berry, is pictured here. And you will see their pictures throughout on the slides where they were involved and performed these studies. So affinity purification, in this case, NS1 was expressed with a tag that was then used to pull down NS1 plus any proteins that were associated with it. And then tandem mass spectrometry was performed to identify the proteins associated with NS1. In this study, 13 of 21 significantly enriched proteins were subunits of the mediator complex. And these are, many of them are here. There are a couple of mediator complex proteins here too. And any of the dots that are in red are significantly enriched in this affinity purification. A nice positive control is that NS1 is the most enriched, which you would expect. So given that what was known about NS1 and was sort of accepted in the field was that it had a very important role in the cytoplasm, but there wasn't really a known role in the nucleus, and it's known that mediator is a nuclear protein. This was both striking and exciting and a little confusing. What did this mean? And so those are that's kind of the trifecta in science for the kind of questions that we like to pursue. So next, we wanted to confirm that NS1 is actually localized in the nucleus for it to be able to bind a mediator, because it was certainly possible that the interaction with mediator was some sort of false positive in that previous assay. And so we took a couple of different approaches. One was Western blotting, where we fractionated cells that were transfected with an NS1 plasmid. And so here you can see, so this is unfractionated cells, the membrane fraction, cytosol, soluble nuclear fraction, and chromatin fraction. And you can see that mediator, as expected, is enriched in the nuclear fractions as a CTCF, which is another chromatin-associated protein, whereas tubulin is not localized in the nucleus, which is as expected. NS1, there is the greatest amount of NS1 is in the cytoplasm, but there's a significant amount also in the nucleus, in the soluble nuclear fraction, as well as associated with the chromatin fraction. Looking using immunofluorescence of infected cells, so either mock infected or RSV infected, we also saw that NS1 and mediator were co-localized in the nucleus, as shown here in green and red, and in these nice graphs that show the overlap a little bit better, because it can be hard to see the dark cells. But you can see that the green and the red are overlapping with the blue, which is DAPI, which stains DNA in the nucleus. So to take a moment and describe the mediator complex, I'm showing this cartoon that depicts the multi-subunit complex. There are approximately 30 subunits in mediator complex. It's enormous. And in this cartoon, it shows a functional depiction of mediator in which the complex here is interacting with a loop of DNA shown here. And the mediator complex is roughly divided into three components: the head, middle, and tail. And some really beautiful recent structural work, and even less recent structural work. A lot of amazing biochemistry has gone into understanding the mediator complex. Has demonstrated that the tail portion of mediator, the subunits in the tail portion, interact with transcription factors and the enhancer of a gene. And then the DNA loops around, and the promoter of the gene that is being regulated by mediator, transcription increasing or decreasing, is then that the promoter portion is bound by the middle portion of the mediator complex. There are additional components of mediator, including the CDK8 kinase module that is also involved in regulation of transcription. And it all becomes quite complex. But the point that I wanted to make with this cartoon is that the mediator complex interacts with transcription factors and RNA polymerase at the promoter to regulate gene expression, and really forms this physical bridge between the two components, the two regulatory elements that are controlling a gene's transcription, the enhancer and the promoter. So as a quick summary of the slides that I've shown so far and an indication of where we're going next, we showed that RSV, NS1, and mediator are both present in the nucleus, in the soluble nuclear and chromatin nuclear fractions. We demonstrated that NS1 and mediator interact in vitro. And as I just showed you in cartoon form, it is known from the literature that mediator complex binds to chromatin at sites of gene regulation. So the next thing that we asked was, does NS1 also bind at gene regulatory sites in the genome? So to address this question, we employed a technique called chromatin immunoprecipitation sequencing, or ChIP-seq, which maps the genomic localization of DNA and chromatin-associated proteins. The way that this works is cells that are, in our case, that had been transfected with an NS1 plasmid are fixed and then sonicated, which means that the DNA is broken up into pieces of uniform size. Those pieces of DNA are then incubated with an antibody against a specific protein. So the important thing about fixing the cells and sonicating the DNA is that when we fix, the proteins are covalently linked, cross-linked to the DNA. And then when the DNA is sonicated and broken up into pieces, those proteins are still bound. So then when we add an antibody to a specific component of that chromatin, the DNA complex, such as an antibody to NS1 or to mediator, the antibody will bind the protein, but the DNA will kind of come along for the ride. We do that incubation for a period of time, usually overnight, and then wash off any of the unbound DNA or protein. So anything that isn't NS1 or mediator is washed away. Those pieces of DNA are then eluted off of the antibody and protein. And then we make a next-gen sequencing library, which I'm sure all of you are quite familiar with. Those libraries are then put on a name your favorite next generation sequencing instrument. And then the data is analyzed. And I've put an example of what ChIP-seq data can look like in this diagram below. And if you can imagine then that the green points are transcription factor 8, transcription factor 1 for transcription factor 2, or in our case, it would be NS1 and mediator, because those are the antibodies that we used. And what these peaks are showing you is where along the genome, this linear expanse here, the line, horizontal line, is indicating a part of the genome, and with genes that are in this region annotated above. The peaks are indicative of the number of sequencing reads that align to that region of the genome. So anywhere where there are green peaks, there was DNA in that region that was bound to, in this case, NS1, that was associated with that piece of DNA. And we were able to determine that using ChIP-seq and downstream analysis. So once we then had the ChIP-seq data for NS1 and mediator, we applied a set of bioinformatic analysis tools that we constructed together in a pipeline. And we identified all of the binding sites for NS1 and mediator across the genome. As shown here in a pie graph, NS1 bound to about 17 or 1800 sites across the genome. And 43% of those, mediator was also bound, which was really a striking enrichment, which further supported the fact that NS1 and mediator that their interaction that we saw in vitro at the beginning of this story may be important in gene regulation. Next, we wanted to know, okay, there's 17 or 1800 binding sites. Do those have a function in the genome? Because a large portion of the genome, we're not really sure what their function is, or they're integrated old viruses or non-coding sites or what have you. So we used a tool that would annotate the genome, the binding sites, NS1 and mediator binding sites for us. And we found that there was significant enrichment of gene regulatory elements in NS1 sites. So promoter and 5' UTR were significantly enriched for NS1 binding. We also then looked at other types of regulatory sites. So promoter and 5' UTR are at the beginning of genes and control transcription of that particular gene. We wanted to know, because of the role of mediator in binding enhancers, is NS1 also bound in enhancers? And so we looked at enhancers in general, and in fact, almost 75% of NS1 sites overlapped enhancers. And then we looked at lung enhancers, since RSV infects lung, and we saw over 90% of NS1 binding sites were in lung gene enhancers. We also wondered whether, so we had already seen that NS1 was bound at the promoters of genes, but are these genes are these just random genes, or are these genes that are actually involved in response to RSV infection? And we saw that, in fact, while there is some overlap of NS1 at promoters, the percentage overlap at differentially expressed genes, so genes that change their expression with RSV infection, is actually more than twice as high for NS1 binding. So that was really exciting to us, and was further evidence that NS1 is having a functional effect on RSV infection at a gene regulatory level. So next, we wanted to evaluate a mutation in NS1, the Y125A mutation, that our collaborators in the Lung Lab had previously demonstrated attenuates NS1 viral replication. And so just to show you where this is located, there's a specific helix in the NS1 that our collaborators identified, and this Y125A is located there. So we looked at RNA expression. These are volcano plots of RNA expression in cells that are infected with wild type RSV or NS1 mutant, the Y125A mutant RSV. And there's, if you're not familiar with volcano plots, log2 fold change is shown on the x-axis, and significance, negative log 10 of the FDR-Q value, which is significance. So significance is increasing on the y-axis, and fold change is either to the right is higher in NS1 or RSV infected cells, and to the left is lower in RSV infected cells. And so as you can see in the NS1 wild-type RSV infection, there are quite a number of immune response and interferon response genes that are upregulated with infection, including IFIT1, IFIT2 and 3, and ISG20. And you can, if you then look over at the NS1Y125A mutant infection, the overall gene expression is significantly reduced. There is much less induced interferon gene expression in the Y125A mutant, which is consistent with attenuated viral replication, as well as reduced expression of the interferon genes with just with the Y125A mutant. So I wanted to call particular attention to IFIT2 and 3, as these were some of the most highly upregulated, and then were their expression was relatively reduced with the Y125A mutant. So we wanted to specifically look at those genes in our ChIP-seq data to understand to understand where NS1 and mediator were binding at those particular very important viral immune response genes. And so again, this is the linear representation of the genome. This is an IFIT locus, so IFIT2, 3, 1, 1B genes. And here's depicted the ChIP-seq data for NS1 and mediator. And the yellow highlights indicate where NS1 or NS1 and mediator were bound. And then below, you can also see that there are small red lines for NS1 binding and small blue lines for mediator binding. You can see then that 3 of the 4 NS1 peaks have binding of co-binding of mediator. One of these is at the promoter of IFIT3, and 3 of the 4 overlie enhancers that are important for expression of the IFIT genes in this locus. So we were very excited to see that we saw an effect on gene expression, and we also saw that there was binding of NS1 and in some cases mediator at the sites of regulatory elements, so promoters and enhancers of the IFIT genes, which are induced and very important for immune response to RSV. So as again, a quick sort of intermediate summary, we showed that NS1 and mediator have overlapping chromatin binding profiles that are enriched in gene regulatory elements, so promoters and enhancers, and immune response genes. And we showed that NS1 mediator target gene expression is reduced with the Y125A RSV infection. So all of this is very exciting and very supportive of our hypothesis regarding NS1's role, but it doesn't directly measure NS1's effect on gene transcription. And so we wanted to test that next. The way we did that was using luciferase reporter assays to measure the impact of different factors, or a variety of different factors you can test, on transcription. So the way that we did that was to take regions of the genome that we had identified that NS1 bound, so those specific areas, the sequence of those areas, the genetic sequence, and we clone those into luciferase plasmids, which are plasmids that have a minimal promoter, and then you clone in your area of interest, and then there's a luciferase enzyme downstream, which is then controlled by the genetic region that you've cloned in. So if the region of the genome that you have included has greater activity in terms of gene transcription, you will see a greater amount of luciferin produced, so more light is produced from those cells. We transfected those plasmids into cells along with an NS1 expression plasmid, and then used a plate reader to tell us how much light was produced. And we found that for these for these regions, so ISRE is a control region, and then we delineated these as IFIT A, B, C, and D, which indicate regions where NS1 was bound in the IFIT locus. So in enhancers or promoters of the IFIT genes. And in each case, the addition, so EV is empty vector, meaning that there was no NS1 introduced into those cells. But when there was NS1 in the blue bars, the transcription from the luciferase vector was reduced. So overall, NS1 had a suppressive effect on transcription from the IFIT locus when these NS1 binding sites were tested. The IFIT B was one exception, but 3 out of the 4, there was a suppressive effect that was significant. So at this point, I wanted to provide some overall conclusions and tell you a little bit about ongoing studies that we have in the lab. So this work, as I mentioned, was published a few years ago. And so you can, if you'd like to find out more, that's in Cell Reports. And this is a graphical abstract depicting kind of a cartoon summary of how we think that RSV and mediator are interacting to control gene expression in the setting of RSV infection. So as I said, we found that NS1 localizes to the nucleus and binds chromatin. Importantly, mediator is not required for NS1 binding. We did find binding sites for NS1 on chromatin that were not associated with mediator sites. But NS1 and mediator co-localize at 43% of NS1 sites, indicating that this is an important interaction. And 35% of genes that had NS1 binding sites were differentially expressed in RSV infection. When we tested the effect of NS1 on gene transcription in those luciferase assays, we found that NS1 decreases reporter transcription at NS1-bound regulatory elements, suggesting that NS1 really does have an effect on gene transcription. And it's not just, its binding at chromatin is not just, sort of we don't know what it's doing or it has no function. It's actually affecting gene transcription. So studies that we have ongoing to continue to understand the role of NS1, we are continuing with the Loon Lab. We're continuing to investigate and elucidate the impact of NS1 mutants on gene regulatory functions, and also to define the role of NS1 in various primary cells from human donors. And finally, I want to acknowledge all the people who did this work. Particularly, I want to acknowledge the Loon Lab and Jinjing Pei, who was the first co-author on the previous publication, along with Nina Berry, who is, since Nina has graduated and is doing a postdoc in another virus in EBV. And Jinjing has a faculty position now and is doing amazing work there. I also want to thank other members of the Payton Lab who helped to get this work done. We've got a previous picture in the green shirts and then a more recent picture at the famous St. Louis Zoo. And of course, I want to acknowledge all of our funding mechanisms. And I also want to acknowledge you and thank you for your time and attention.